Antigen antibody reactions. Antigen antibody reactions general features. Antigen combines with a specific antibody in an observable manner, and the reactions between antigen and antibody is specific. These antigen antibody reactions in vitro are known as a serological test. Uses in the in the body or in vivo, it forms the basis of immunity against infectious diseases. It may lead to tissue injury in some hypersensitive reactions and autoimmune diseases. In the laboratory or in vitro, for diagnosis of infections, detection and quantitation of either antigen or antibody. Characteristics: the reaction is specific. An antigen combines only with the homologous antibody and vice versa. The entire molecules of antigen and antibody react and not the fragmented. The reaction is firm but reversible. The firmness of combination depends on the affinity and avidity. The affinity is defined as the intensity of attraction between antigen and antibody molecules. The avidity is the binding strength after the formation of antigen-antibody complexes. The antigen-antibody can combine in varying proportions. The antibody are generally bivalent. Although IgM may have 5 or more combining sites, the antigen may have valency up to hundreds. A single antibody can cause different types of antigen-antibody reactions, and a single antigen can stimulate productions of different classes of immunoglobulin, which differs in their reaction capacities as well as in their properties. Types of antigen-antibody reactions. Precipitation reactions. When a soluble antigen reacts with the antibody in the presence of electrolytes NaCl at an optimal temperature and pH, the antigen-antibody complex forms an insoluble precipitate, and it is called as a precipitation. The precipitation may occur in liquid medium or in gel such as agar, agarose, and polyacrylamide. The precipitation usually sediment at the bottom of the tube. Flocculation. When instead of sedimenting, the precipitate is suspended as floccules, the reaction is called flocculation. This is a modified form of precipitation. A serological test, video oral routine used for diagnosis of syphilis, is one example of flocculation test. Video oral slide test is used to perform venereal disease research laboratory test. Prozone phenomena. The precipitation occurs when the antigen and the antibody are present in the optimal or equivalent proportions. The absence of precipitation, false negative, is present of excess antibody is known as a prozone phenomena. These sera with the excess antibody should be diluted further to get the proper result of precipitation. Mechanism: precipitation. Lactic hypothesis. The application of precipitation reactions. The application of bacteria. Example: ancestral grouping of streptococci. Detection of antibody for diagnostic purpose. VDRL in syphilis. Forensic application in identification of human blood and seminal stain to standardize toxin and antitoxin. Type of precipitation reactions. Ring test. Antigen solution is layered over an antiserum in a narrow tube. A precipitate ring appears at the junction of two liquid. Example is streptococcal group by Lanceville technique. The flocculation test. It can be done in slide and in tube. The VDRL test for detection of antibody against syphilis is an example of a slide agglutination test. Cons test tube flocculation was done previously for diagnosis of syphilis. Immunodiffusion test. These are precipitation tests in gel. The immunodiffusion is generally done in 1% agarose gel. Various immunodiffusion tests. Single diffusion in one dimension, Audin procedure. Double diffusion in one dimension, Oakley full Harpy procedure. Single diffusion in two dimension, radial immunodiffusion. The antiserum antibody is incorporated in agar gel on slide or petri dish. The wells are cut on the surface of the gel. Antigen is added on the well to the well. Antigen diffuses radially, and a ring-shaped band of precipitate halos are formed around the well. The diameter of the ring is directly proportional to the concentration of the antigen. Uses: it has been widely employed for estimation of immunoglobulin classes IgG, IgM, IgA in sera. Double diffusion in two dimension, Outerloni procedure. Immunoelectrophoresis. Immunoelectrophoresis combines electrophoresis and immunodiffusion. By this technique, a number of antigens can be identified in human sera. It is particularly useful for detection of normal and abnormal serum proteins like myeloma protein. Electroimmunodiffusion. The immunodiffusion can be speed up if antigen and antibody are driven by electricity. It is combination of electrophoresis and diffusion. Of this method, counter immunoelectrophoresis and rocket electrophores are used frequently. Counter immunoelectrophores, counter current immunoelectrophores, CIE or CIEP. This test is based on the movement of antigen and antibody in opposite direction. This is performed on a glass slide layer with agarose gel, and wells are cut on this surface. One well is filled with antigen and other with antibody. Electric current is passed through the gel. Antigen and antibody moves towards each other, resulting in precipitation line. It takes about 30 minutes and is more sensitive than standard immunodiffusion technique. It is applied for detecting a hepatitis B antigen and antibody. Antigen of cryptococcus in cerebrospinal fluid and in number of other diseases. Rocket electrophores. This technique is mainly applied for quantitation of antigen. The antiserum of antigen to be quantitated is mixed with agarose and gelled on the glass slide. The well or punched in the set gel and filled with increasing concentration of antigen. It is then electrophorus. The precipitation is formed in the shape of cone-like structure. Appearance of rocket. The length of this rocket-like structure corresponds to the concentration of antigen. Agglutination. It is an antigen antibody reaction in which a particulate antigen combines with its antibody in the presence of electrolyte at an optimal temperature and pH, resulting in clumping of particles. It differs from precipitation, in which the soluble antigen is present in contrast to particulate antigen of agglutination. The agglutination is more sensitive than precipitation for the detection of antibody. The agglutination reaction takes place better with IgM antibody than with IgG antibody. The principle governing agglutination are the same as precipitation. Agglutination occurs when antigen and antibody are present in optimal proportion. The lactic formation hypothesis holds good for agglutination too. Type of agglutination reactions. Slide agglutination. Uniform suspension of antigen is made in a drop of saline on a slide, and a drop of the appropriate antiserum is added. Clumping occurs instantly or within second when agglutination test is positive. Uses. It is a routine procedure to identify the bacterial strain isolated from a clinical specimen. One example is to identify Salmonella species. It is also used for blood grouping and cross matching. Tube agglutination. This is a standard quantitative method for determination of antibody. Uses serological diagnosis. Entering fever, viral typhus fever, wheel flex reaction, infectious mononucleus pulmonal test, and brucellosis. For diagnosis of primary atypical pneumonia, streptococcus MG agglutination test. Heterophil agglutination test. The heterophil antibody has a property to react with the microorganisms or cells of unrelated species due to the common antigenic sharing. Examples of heterophil agglutination test includes whale flex reaction, pulmonal test, streptococcus MG agglutination test, passive agglutination test. A precipitation reactions can be converted into agglutination test by attaching a soluble antigen to the surface of carrier particles such as latex particles, bentonite, and red blood cells. Such tests are called passive agglutination test. The conversion is done because agglutination reactions are more sensitive for detection of antibody. When instead of antigen, the antibody is observed on the carrier particles for a estimation of antigen, it is known as a reverse passive agglutination. The latex agglutination test. The polystyrene latex particles are widely employed to observe several types of antigen. Latex particles can also be coated with antibody for detection of antigen. These tests are used for detection of hepatitis B, ASO, CRP, RA factor, HCG, bacterial type in Neisseria meningitis, and many other antigen. Heme agglutination test. The erythrocyte sensitizes with antigen are used for detection of antibody. Co-agglutination. The principle of this test is shown in the figure. This test is used for detection of bacterial antigen in the blood, urine, and CSF. Neisseria gonorrhea, streptococcus pyogenes, and hemophilus influenza antigen can be detected by this method. Complement fixation. Antigen antibody complexes has ability to fix complement. This reaction has no visible effect. To detect the fixation of complement, an indicator system consisting of sheep erythrocyte coated with a amboceptor rabbit antibody to sheep erythrocyte is used. The complement can lyse these erythrocyte coated with antibody. If complement is fixed and utilizes in the antigen antibody reaction, there is no free complement to act on the indicator system, and hence no lysis of erythrocyte, which indicates the positive complement fixation test. Lysis of erythrocyte indicates that complement was not fixed in the first step, and therefore the serum is negative for antibody. negative CFT. Neutralization test. The bacterial exotoxin are capable of producing neutralizing antitoxins, which plays a role in protection against disease such as diphtheria and tetanus. Viruses may also neutralize by their antibody, and these are named as a virus neutralization test. Some examples of neutralization test are anti-streptomycin ASO test, virus neutralization test, and Nagler reactions. Immunofluorescence. Fluorescence is a property of certain dyes which absorbs rays of one particular wavelength ultraviolet and emits the rays with different wavelength visible light. The fluorescent dyes can be conjugated to antibody, and these label antibody can be used to detect antigen in tissue. The commonly used fluorescent dye are fluorescein isothiocyanate, and lysamine rhodamine exhibit the blue, green, and orange red fluorescence respectively. The immunofluorescence test is of two types: direct immunofluorescence and indirect immunofluorescence. The principles of direct and indirect immunofluorescence are shown in the figure. The test is commonly employed for protection of bacteria, viruses, and other antigen in the blood, CSF, urine, feces, tissues, and other specimens. Direct immunofluorescence test is a sensitive method for detection of a rabies virus antigen in brain smear. Indirect immunofluorescence FDA antibody test is used to detect antibody in the serum of syphilis patient. Radioimmunization is based on the competition for a fixed amount of specific antibody between known radio labeled antigen and unknown unlabeled test antigen. This competition is determined by the level of test antigen present in the reacting system. After antigen antibody reaction, the antigen is separated into the free and bound fraction, and their radioactivity is measured. The concentration of the test unlabeled antigen is calculated from the ratio of the bound and total antigen label using a reference curve. This test can detect antigen up to picogram, 10 to the power of minus 12 gram quantities. Enzyme link immunosorbent assay. ELISA has been applied widely for the detection of varieties of antibody and antigen. It is simple and nearly sensitive as radioimmunization. The principle of ELISA is almost same as that of immunofluorescence. The only difference being an enzyme is used instead of fluorescent dye. The enzyme acts on substrate to produce a color in a positive test. The test for specific immunoglobulin classes IgG ELISA are also available. The principle of sandwich ELISA, indirect ELISA, and competitive ELISA are shown. Uses of ELISA: detection of HIV antibody in serum, detection of mycobacterium antibody in tuberculosis, detection of rotavirus in feces, detection of hepatitis B viruses in serum, detection of enterotoxin of E. coli in feces. Immunochromatography. The immunochromatography or lateral flow immunoassay is one of the most popular form of radioimmunoassay. It can detect both antigen and antibody. It can be complete within 30 minutes. It is a strip-based test. The strip contains a chromatographic pad and 3-zone sample pad, the conjugate pad, and capture line. The conjugate pad may be having a colloidal gold dye or latex beads as conjugate with which produces a signal. The specimen is applied to the sample pad and flow laterally by capillary action. Upon reacting the conjugate pad, it may bind to conjugate if antigen or antibody present in the specimen and forms antigen antibody complex. This complex then flow laterally to reach the capillary line. Here it is captured by antigen or secondary antibody present in the capture line. The presence of color line is positive. There is a positive control line also to check that the test was properly performed.